today, I'm very pleased to have Bob Gross here with us. Bob is an MD, PhD, trained at the Albert Einstein College of Medicine, and is now a clinician scientist at Emory University, where he's the MBNA Bauman Professor in Neurosurgery and Neurology. He's the president of the American Stereotactic and Functional Neurosurgery Society. And at Emory is the co-founder and director of the Emory Neuromodulation and Technology Innovation Center in TICE, which is a very unique combination of clinicians, scientists, and engineers seeking new treatments, new therapies for diseases. I encourage you to check out their website, do a Google of InTICE and Emory, and find out what they are all about. They run some really interesting events where we bring clinicians and engineers and scientists together at that intersection. Today we'll be hearing about the work in his lab, which is the Translational Neuroengineering Lab at Emory University. I'm very pleased to have him here. If you're joining us remotely, I encourage you, please mute your microphone so I can hear someone fiddling around in the background. Thank you very much. Bob, thanks for joining us. Welcome.
Thanks. My pleasure. Thanks for inviting me, Chris. All right. Well, I'm not an engineer, even though I'm program faculty in engineering and PhD advisor to a number of bona fide engineers. But I was majoring in engineering for the first semester of college. So you eventually return to your roots. I found my way from there to computer science, and then ultimately to neuroscience, which is where I ended up. But it's interesting how things go around and come around. So I'll talk to you about neurotechnology, really at the interface of science and medicine. So that's really where I focus. Anything that I do in a research project has to have an application. As you will see, some of the applications are immediate now. Some of the applications are quite far off in the future, if at all. But we always have that angle of approach. It's fine for other people to do discovery science and even to do, of course, engineering that doesn't have a clinical application, but that's the unique focus of my lab. I do have some disclosures in respect to some of the things I'm going to talk about really clinically, and this reflects the fact that if you are going to deal with technology at the interface of medicine, you will be dealing with commercial entities. Commercial entities in our capitalist society and economy are important because they drive the application. There's the saying, many of you might have heard, no margin, no mission. And so we have to work very closely with them, and so I work as a consultant for some of these. I don't have any actual ownership in any of the companies, but in particular, I'll talk about some technology from Medtronic, MRI interventions, and some of these others, and NeuroPace. So the focus of my applications today are epilepsy. Epilepsy is characterized by recurrent seizures, where you are seized, ultimately named after being seized by the devil, essentially. So we still use that arcane terminology. And when you have recurrent seizures, you have epilepsy. And it affects a lot of people. About 1 in 100 people have epilepsy per se. It involves synchronous discharges of neurons throughout the brain. And we see that on an electroencephalogram, which is recording electrical activity at the surface. And usually it sort of marches off in this fashion where it looks pretty flat. It's not completely flat, or you wouldn't be doing very well. But it has these slow oscillations when all of a sudden large populations of neurons begin to fire together. And of course, it's not every neuron in your brain that's firing together. It's just a very small subset. But if you imagine in this room, if only 3 people started whispering at the same time the same thing, then it actually would be quite distracting to the rest of the people in the room. And if 15 people out of the 100 in the room started whispering the same thing, I wouldn't be able to continue to talk over that cacophony. So it doesn't take much synchronous behavior to have an epileptic seizure, and that may be one of the problems in treating it. And that is that we have to identify these small populations of neurons that are firing together. The epidemiology is such that actually the cumulative incidence or the risk per person is 2 to 4%. So if you think if there's 100 people in this room, 4% or 4 people in this room will either have had or, and I won't ask who's had a seizure, but either have had or will have a seizure during their life, or have epilepsy during their lifetime. And the risk of having at least one seizure is 10%. So 10 people in this room will have a seizure at some point during their life. We call it an emergent property of the nervous system. So in doing the things that you're trying to do perhaps right now, which is learning and remembering what we're doing, that sets the system up to have this abnormal types of overly synchronous connection between parts of the brain. Now, 2/3 of the patients are actually treated with medicines and that have epilepsy and don't continue to have seizures upon taking medication. But 1/3 of the patients that we see have medically refractory seizures. So 30%, 30 to 33% of the people that have epilepsy and are treated with anti-epileptic drugs will continue to have seizures despite that. And that has not been changed since the very first anti-epileptic drug, potassium bromide, was discovered, close to 100 years ago. So despite decades of targeted drug discovery, we have not moved the marker on that. So what do we do in people that have drug-resistant epilepsy? Well, that's where I come in. Okay, so we have surgical treatments, and this has been going on really since the dawn of the field of neurosurgery in the late 19th century, really in the 1890s. And we've been removing large chunks of the brain. This shows removing part of the temporal lobe and the medial temporal structures, or the hippocampus over here. And this shows a more selective approach to that. And you can see we remove large chunks of brain in order to treat this problem. And you can imagine that something like this might have some adverse effects on patients' functioning. Okay? Strikingly, sometimes it doesn't, but that's really where what we're trying to deal with is this problem. And sometimes we do things even more radical. This is called a hemispherectomy. So back in the 1930s, it was discovered that if you remove a whole hemisphere of the brain and the epilepsy is located in that you can stop seizures. Okay, that's pretty dramatic approach. Nowadays, we do things where we leave the hemisphere in and we just disconnect it, and sometimes even more subtle, where we just, every place has a black, this is the ventricle, but every place has a black mark, is cutting through the pathways that connect the epileptic area to the rest of the brain, so-called functional hemispherotomy. But that's pretty radical. And we do things like cutting through the corpus callosum, that highway that, as you know, connects one side to the other. So a corpus callosotomy can divide the brain and disrupt the synchrony of bilateral discharges and be a reasonably effective operation, although with adverse effects like the disconnection syndrome, which is actually what drew me into neuroscience when I was an undergraduate, as many of you are, was being very interested in hemispheric asymmetry. That's what drew me into this field to begin with. So this is very effective, and this shows you a randomized single-blind controlled trial where patients were randomized to just continuing medical treatment and looking at the percentage without seizures. And if you take this group, only 8% of the patients became seizure-free with continued medical treatment, whereas 64%, but the details are not important, became seizure-free with a temporal lobectomy. So that's pretty effective. That's good news for patients. I can tell you 2/3 of the patients will be seizure-free. And that's just the temporal lobe, and that pertains to all operations we do, basically. If you look at sort of meta-analyses or systematic reviews, 65% become seizure-free, and 25% become seizure-free and off their anti-epileptic drugs, which is a cure. So that's really good news on the one hand. On the other hand, it creates problems, as I said. Removing large chunks of brain can have adverse effects. So this shows the picture of after we remove this medial temporal structure, that there can be white matter changes in the areas that are in the vicinity of the places we're operating on. And if you actually look at the amount of what we call collateral damage, off-target effects, the more collateral damage you have, the more loss of function you have. And the less collateral damage you have, the more gain of function you have, actually, from rendering patients seizure-free. So this is at a cost. And these, and what we really are trying to do is trying to maximize the benefits in anything that we do. And I told you we're at about 65%. That's over here. Well, we want to get up over here. And every time we take out more brain tissue, as we take more brain tissue out, we get to more deficits. So this is a patient that has benefits, but that has deficits. This is a patient that has no benefits and no deficits. And somewhere in between is the sweet spot where you get benefits and minimize the deficits. And that's really what we're trying to do by engineering the system a little bit more. So minimize off-target collateral damage. This shows someone going after a fly and ruining the room. So that's a depiction of the collateral damage. And we want to optimize the on-target therapeutic effects. And we also want to decrease the pain and suffering that goes along with surgery. Because as you can imagine, when I take out a large chunk of the brain, there's a lot of pain from the surgery. People take a long time to get back to work. They stay in the hospital. And because of that pain and suffering, a lot of patients never come forward for surgery. So we are very, very underpenetrated in the market of patients that could be helped with surgery, because we might say, well, I know you're having seizures despite your medicine. If I do an operation, they say, no, I want nothing to do with that. I'm not going to have brain surgery. Or, even perhaps more importantly, the referring doctors are very scared of surgery. So a patient might come in to see their doctor and say, hey, I saw a report on CNN about how this could help brain surgery. And they say, no. People end up as vegetables. Next question, I have a new drug for you. So this pain and suffering and the collateral damage decreases the accessibility of patients to getting treated. And people die from epilepsy every day. So the first engineering advance I'll talk about is one that was not my own, but because I was really at the forefront of dealing with new types of technology, I was one of the earliest adopters of this therapy, called laser interstitial thermal therapy. So there's a very long history of using lasers in neurosurgery. It dates back to the invention of the laser back in the 1950s, and these very large and cumbersome devices. Much of this work was done at Bell Labs, and the Nobel Prize was given to the first functioning laser. And then there was, as you'll see, becomes important, the invention of the light-emitting diode, actually, and the LED laser source is very important in making these available for use. And you can see these early lasers were not particularly elegant devices, and they also were not very minimally invasive. This is hardly what we would call minimally invasive neurosurgery, and you can see there's a large amount of damage. And the original lasers were actually developed as cutting instruments. Okay, they caused immediate vaporization of tissue. And because of the cumbersomeness of this and the fact that they were only used as cutting tools, and we have other cutting tools without having to deal with something like this, there was a very slow adoption. We had lasers when I was a resident back in the 1990s, but I know we had them. I can't say I ever used one during a case because they were cumbersome and not particularly useful. But they have the potential for precise delivery of thermal energy, and with the advancement in certain types of visualization technology, they ultimately became more useful. So the idea of using a laser instead of as a cutting tool, but as an interstitial tool, was developed in the 1980s, some 30 years after their invention. Okay, the idea here was to put laser light into the tissue and release photons at a much lower energy level, where those photons would cause localized heating. And that heating can cause a thermocoagulative necrosis or damage. So this idea was born in the 1980s. And as compared to the photomechanical damage that goes with these short pulse, high energy laser exposures that cause vaporization and instant perforation, This causes photothermal damage, which is a medium exposure level, continuous wave laser, and tissue coagulation thermal denaturation. And this looks quite a bit different than the other type of picture I showed you, okay, where there's an area which has lost its blood supply and where the tissues have been heated to the point of enough destruction that they will ultimately die. So rejoining the timeline here, this was used in the 1980s to treat certain deep brain gliomas, but it wasn't particularly useful because you couldn't tell how hot you were heating the tissue. Okay, if you're doing what's called a radiofrequency ablation, which uses a metal probe, you can put a thermocouple in there and monitor the temperature. And temperature is critical. But until we could do that with lit, it didn't become useful. But there were certain other technical advances, engineering advances, that occurred in the 1990s, which is the initial use of MRI. And you could observe the effect of the laser, but not immediately. So afterwards, you could see the destruction, but you couldn't see it immediately while it was going on. But in 1995 now, Toshiba released a paper demonstrating the ability to observe temperature based on the phase change of water molecules in the tissue. So you could actually use the phase change in the MRI as a thermometer, and you can use the MRI as a thermometer. And this married up very nicely with this laser interstitial thermal therapy, and this shows what that looks like. So the system can use MRI sequences which have high susceptibility. So not your typical one just for looking at structure, which requires a lot of fine-grained detail, but the type of sequence you would use for an fMRI with high temporal resolution. And you can, in a linear way, exactly relate the change in temperature to the change in the phase of the protons during heating. So this very nice and tight linear relationship allows you to look at change of phase and read a temperature change. And this is what the imaging looks like. So it's obviously not very good for structural imaging appearances. You're looking at a phase map here. So it encodes all phase changes by using warm and cold colors. So this is a heat map of a heat map. That's a joke I always tell. I saw one guy chuckle over there. At least in a crowd of engineers, some people chuckle with that. So, and so this is what it looks like as put together ultimately by the company that sells this. And this is a thermal map right over here. And this is, now, just looking at a thermal map, I can't tell exactly what's happening. But if I overlay this on top of a structural image, and they're co-registered in MRI space, so there's no movement allowed from this thermal map to the archival or historical imaging set, then I can see that this damage is happening to this area of the brain. And now, I'm using the temperature change, but if I, as a surgeon, am sitting there and reading the temperature of the brain, I can't tell what's happening. I can just tell that an area has gotten to 60 degrees or 40 degrees. And what I want to know is what has been damaged. Well, it turns out that the chance of being damaged is a function of two things. One is temperature and time. The longer you have something at a particular temperature, the more chance it will be damaged. And so we're looking at the area under the time temperature curve, and that is quantified by this integral, which I'm sure some of you know what that means. This basically looks at the area under the time temperature curve. And what we do is, this is the actual live thermal map, and I can see that this area is being heated up, but by calculating the Arrhenius model, we can depict the areas that have been fated to be damaged because they've reached a critical amount of time temperature, and that grows in front of us. So I can see the area that has been fated to be damaged here by this damage zone estimate. This takes this advance and then couples it together with laser interstitial thermal therapy, first done in the late 1990s, and allows us to develop this MR thermal imaging and couple it together. And this is what really drove the whole field forward. So these are the critical advances that have moved this from a therapy which was in the 1980s not something usable and just tried on a couple of patients with gliomas to something that we're using every day now. Now the diode laser was, I told you about the LED, that was really critical because you have to be able to wheel these things around. I'm bringing this thing down to the MRI suite. I can't wheel that huge laser down to the MRI suite. So that was an important advance. Having cooled catheters, certain automation, and these real-time damage zone estimates are critical to this therapy. So in 1997, the late 1990s, Ashok Gowda and Roger McNichols, who are two biomedical engineers working down in Houston, they both trained at Rice. They put their heads together and they founded a company called Biotex based on this application. They eventually spun this off as Visual Lace. Clever name, laser, visualize it. So Visual Lace, and a Visual Lace was eventually purchased by Medtronic. And the first procedure using this turnkey approach where the laser, this is the diode laser over here, this is the imaging, or the computer display, the computer down over there, and there's a peristaltic pump to cool. And this was eventually used in 2006. It was FDA cleared by a 510K mechanism. Someone wants to ask me about later on, I can talk about that. And the first neurosurgical procedure was done in 2008 in the United States. And the first time it was used for epilepsy was by a friend of mine, Dan Curry, down in Houston in 2010. And we did our first procedure in 2010 as well. This is what it looks like. So the laser is introduced through a fiber optic. And this is done by stereotactic technique. And you can see the laser at the end of this. The fiber optic comes down the catheter, and then there's a diffusing tip at the end, which diffuses the laser light into the tissue around it, where the photons bounce around and cause localized heating by their interaction with macromolecules that absorb at that particular wavelength of light. So it's a very particular wavelength of light that's used. And this is how the whole thing works, in that I told you about vaporization of tissue at greater than 100 degrees. At 43 degrees, nothing happens. But somewhere in between, you get this time-dependent thermal damage in this area between 45 and 60 degrees. So this, again, is what the thermal map looks like, the damage zone estimate. This is, I already showed you what that looks like. This shows a very nice screen for tracking these. And this shows what it looks like in actual practice. Okay, this is a coronal view, and this is an axial view. And you can see the thermal map. You see the fiber optic being passed into the hippocampus. And then you see the irreversible damage zone beginning to accrue in the hippocampus. And I can follow that. And when I've said enough is enough, and I can also make sure that there's no damage to surrounding structures like the visual pathways over here or the brainstem over here, because I can directly visualize them. I then pull the catheter back. And then you may want to... Yeah, someone on the remote side, if you can turn your microphones off, please. You are echoing over here. Thank you. I think we made some progress. Great. Okay. Now this is introduced by various technologies. Some of these are all stereotactic tools that we use to put these things in. And this is the way the system was designed, and this is how we use it. So this is another nicely engineered platform. So this is called an MRI targeting platform, and this allows us to finally adjust this in the MRI scanner. So the way I do this procedure is I bring the patient directly to the MRI scanner, and we're able to do the surgery in the MRI. And we attach this frame to the patient's head, and this is basically a gun sight. So this has a gadolinium containing tube over here, and I can see based on that gadolinium tube where it is virtually projecting, and then I can adjust that using these dials to adjust the azimuth and declination and the translation until I get it just on target, and then I can introduce this fiber optic right into the brain through that technique. And it works out quite nicely. This shows what the damage zone looks like. This shows what the actual damage is, and these overlap very, very nicely. So the damage zone very nicely predicts the actual damage area. And so we have looked at the outcome of this, and this is a paper we published earlier this year with the help of my graduate student, Matt Stern, who's sitting in this room. Matt, raise your hand. So worked very hard on this. And what we showed was that about 60% of patients have become seizure-free for at least a year using this technique. So it's about the same level as we were seeing with open temporal surgery. Well, have we moved the marker as far as that collateral damage that I told you about? So this shows the work of Dan Drain in our group, looking at deficits in basically object recognition and naming. And if you go down over here, you've had a decline in object recognition. And this population are all patients that have had open temporal lobe surgery, the kind that I showed you earlier where we actually removed the temporal lobe. So all of those patients experience declines in object recognition and naming, whereas the patients that have had laser ablation experience no decline and in fact experience a recovery of some of those functions. So we are able to get a reasonable degree of effectiveness and decrease the collateral damage. And this has substantially increased the acceptance of this operation. Whereas previously I would see patients in my office and I would propose doing a temporal lobectomy, and they would say, many of them would say, no thank you, I'm not interested, and they would continue to experience seizures, whereas now nobody is refusing that surgery anymore. And that's actually, from our neurologists, we don't have any patients that are not interested in surgery because of the laser. So we have a lot of different things that cause epilepsy aside from temporal lobe epilepsy. There's tumors, gangliogliomas, there's cavernous malformations, hamartomas, malformations of cortical development, and this is a useful tool for all of these actually. So this shows a very deep-seated hypothalamic hamartoma, which you can imagine if I were to do this in an open surgery would be quite difficult to get to and associated with a lot of potential for injury. And this shows the damage zone, the thermal map and the damage zone associated with that. This shows other types of deep-seated lesions. So once you have a hammer, a lot of things look like nails, and we are in the process of spending a lot of time on exploring other usefulness of this device. I showed you a picture earlier of the callosotomy of dividing the corpus callosum. We can even use this for dividing the corpus callosum. So this shows a patient with 5 different bolts in their head for introducing the laser over 5 different places. And you can see that we're able to very nicely ablate and destroy the whole corpus callosum. And we've done this on about 13 patients now. And it also is benefiting them as well. Well, what do you do, though, however, when a destructive procedure is just not the right operation for a patient? So there are patients, for example, that have an onset zone right in their motor cortex region of their dominant hand. All right? Well, there's no chance of developing a therapeutic window. Damage that gets rid of the epilepsy also gets rid of the function. So no therapeutic window. What do we do in those instances? So this is on to the second engineering advance, so new approaches to neurostimulation. Well, this is a beautifully engineered device that was developed in part by intellectual property developed here by a guy named Brian Litt, who's now a professor of biomedical engineering and a neurologist up at University of Pennsylvania. And while he was here, he helped to develop the idea that if you run an EEG and you detect the seizure onset, you can stimulate that area electrically through a pulse generator generating an electrical signal in the brain. And that will actually suppress the seizures. So there are certain seizures that if you hit them hard just at the right moment, you can actually break the seizure. And so this was encapsulated into a device called a responsive neurostimulator that runs a continuous EEG. And this is what Brian's contribution was, the closed loop feedback activation of the unit in response to detecting a seizure. And actually what's nice is that the patient can this will store that information, and the patient uploads the information to the internet, where I can then go online and see if the patient's had any seizures over the last 24 hours and how effective this device is. So this is a skull-mounted device. It has to be skull-mounted because it's recording neural signals, and you need to have the head stage pretty close to the electrode to minimize the noise floor. And this shows electrodes in this particular instance in the hippocampus, one on each side. And this shows a patient of mine that has electrodes that are implanted into the motor cortex, and this is just such a patient as I just described. She had onset in her.
Sorry to interrupt.
No.
Continue.
Okay. Did you mute me or something?
Someone muted you.
Oh, thanks, Chris. I'll go back and redo all that. So this shows a patient with electrodes implanted into her motor cortex and the device implanted in the skull. And she has had no complex partial or generalized seizures since the surgery and a reduced frequency of these focal motor and sensory events. So she's had a nice response, but she's not seizure-free completely. And patients want to be seizure-free. So this is a randomized, double-blind, controlled trial of sham stimulation, not turned on, and stimulation that was turned on, and this showed about a 35 to 40% decrease in seizures in this population of patients, and this was sufficient to garner approval by the FDA for this device. This shows the percent change in seizures from their pre-implant period, and you can see that some patients have no response or very little response. Few patients actually got worse. Most patients got better. This is 100% response over here in a handful of patients. So about 10% of patients became seizure-free, with the majority of patients, 65%, having a reasonable greater than 50% benefit. So if you have an onset in an area that cannot be removed, and you are not someone that I can cure of their seizures, this is a great device. Except that when I tell people that the happy news is you get about a 65% reduction in their seizures, except for a handful of patients, they say, that's great, I'd like 100% please. So we have to try to continue to move the marker. This is great, but we have to keep going forward. So how can we increase the effectiveness of neuromodulation for epilepsy? So that's been the focus of my group. So this is a multi-electrode array on which is cultured neurons, cortical neurons. And this was work done by Steve Potter, who was faculty here at Georgia Tech up until just a couple of years ago. And this shows what happens when you put neurons on these microelectrode arrays. So this is stacked 64 channels, okay? And you can see that after a time, this shows the after a time period, that they start to fire very synchronous bursts of activity. So that when they start to fire, they entrain each other and they fire together. So if these were firing all separately, you wouldn't actually see anything here, really. But the fact that you see these big black marks shows that these are synchronous populations of neurons firing together. And what Steve showed was that if you stimulate them all synchronously at a low frequency, you can actually entrain the whole culture together. So it actually makes things worse. But as you increase the signal size and as you increase the number of electrodes firing, you can begin to back this off so that you get basically close to this situation. And this is with single site stimulation, when you just stimulate one site. And then he looked at what happens if you stimulate across all the arrays, all the electrodes, across all 64. So in a distributed multi-site fashion to basically try to break up the activity. And he did this in a feedback-controlled way where he actually used as a biomarker the spikes per second array-wide. So we're trying to get rid of these synchronous spikes. We can look and see when the synchronous spikes go away, but you can also use this surrogate. How many spikes per second are happening array-wide? And as you pass the electricity into it, more and more electricity, you're getting more and more spikes elicited. And that is a surrogate for actually decreasing the amount of synchronous behavior as well. So as you turn it up higher to 500 spikes per second array-wide, you're able to get rid of this synchronous activity. So you can't see these action potentials here, but the fact that there are no big black bands tells me that there's no synchronous behavior. So Steve was able to eliminate synchronized bursting in these cultures by using a surrogate, spikes per second array-wide, as he delivered multi-electrode, multi-site distributed stimulation. So when we started working together, we said, well, how can we get this to the bedside? All right? How can we move from the bench to the bedside. Well, it'd be kind of hard to go into a bunch of venture capitalists and tell them that, hey, I've got this thing and it works great. Now I want to stick this thing into someone's head. Okay, so you need a translational pathway. And so we started out with the rat. This is the work of John Ralston and Sharanya Desai. Did this in the group. John Ralston is now a neurosurgeon. He was an MD, PhD, got his PhD here. He's now a neurosurgeon at University of Utah. And Sharanya Desai is a PhD, and she's now working for that company NeuroPace that made the responsive neurostimulator I just showed you before that was implanted in the head. All right, and during their thesis work, they made these multi-electrode arrays. We put them into the hippocampus in a rat, and we passed patterned theta frequency stimulation, and we hypothesized this would suppress the seizures. We used a rat model called the tetanus toxin model, where we infused tetanus toxin, which changes the ratio of excitation to inhibition in the favor of the former, and leads to seizures in close to 100% of the rats that we do this to. So that's our animal model. And what we did was we looked at pre-stimulation and then stimulation and then post-stimulation and looked to see what the effects of stimulation were. And we did different types of approaches to this, where this was continuously done versus being 2 minutes on, 2 minutes off during that period of time. And we also looked at a macroelectrode, a big electrode like the electrode I showed you before when putting in that responsive neurostimulator. So does this perform better than a macroelectrode using these microelectrode arrays? So when we pass synchronous pulses in, we can entrain the whole, or rather, this is what the synchronous pulses look like. We look at different types of pulses. And this is the data. And it shows that when we stimulate using asynchronous, like Steve did, okay, not synchronized across the whole population, continuous stimulation or intermittent stimulation in the theta range, we were able to suppress seizures during the stimulation period as compared to before and after stimulation. And if we look at various other synchronous populations and different approaches, we were unable to get that same result. So that supports the idea that this distributed multi-electrode asynchronous stimulation can actually suppress seizures. I didn't show you one of the control conditions before was with a macroelectrode. We couldn't get the same effects with the macroelectrode. All right, so now we're not going from the bench to the bedside, we're going from the rat to the bedside. And can we go right from a rat to a human? That's also a very difficult jump to do, especially because we're not only trying to show in a much larger species that it works, but that type of array that we use in a rat, using a neuronexus probe with 15 spikes, putting that into the hippocampus deep in the brain is not something you can do. So we really need to translate both the finding as well as the technology and do it in a fashion that is amenable to a large primate. And so the pathway for us is through the non-human primate. And I know that last week Dr. Devernyas was here, and she talked about some of the non-human primate work that we're doing. And she's one of the principal investigators on this with Babak Mahmoudi, a biomedical engineer working with us, and neurologist, and a PhD in my lab. And this is the scheme behind what's called a UG3-UH3 grant from the NIH, from the Brain Initiative, that was given to us a couple of years ago to translate this work. So what does UG3-UH3 stand for? The first question that I asked when I got on the phone with the program officer after we were told of our score and we wanted to talk about the milestones for this grant, I said to them, the first question I have for you is, what does UG3-UH3 stand for? And the answer was, we have no idea. So, and what's really, I was especially curious about it because we applied for this twice. The first time we applied for it, it was called a UH2-UH3, and then they felt the need to change it to UG3-UH3 without even knowing what it stood for. So the NIH works in mysterious ways. But the UG3 refers to what's called the preclinical part, and the UH3 is the clinical part. So maybe the H stands for humans, and then the G stands for non-humans. I'm not, gonanimals, something like that. So this is our scheme for getting this to humans. So the second part of the grant is to go into humans with a device. And the first part of the grant is to translate that into a non-human primate model. So we start, here's the rat. These are the studies we've done already. And first we have to have a data acquisition system that's appropriate. And instead of challenging ourselves with having the translatable data acquisition system to begin with, we're going to use just a typical black rock, and we started this work about a year or so ago, and develop the type of electrodes that we need for the humans, and then ultimately go into a system called an RC+S, which is a type of deep brain stimulator system. A component of this is to develop our own surrogate. So I told you that spikes per second array wide was a surrogate that allowed Dr. Potter to tune his device so that it could achieve the clinical result of getting rid of the burst firing. So we have the same problem. Call this the burst firing over here, and we're looking for a biomarker of response like spikes per second array wide. It takes weeks and years to evaluate the clinical response of these types of things, whereas using a biomarker allows us to figure it out in anywhere from microseconds to at least months. And so one of the components of the grant is to look for biomarkers of the response. This is just some of the parameterization of how we're going to do it. This shows the development of the electrodes, the type of electrodes we're developing. These are our equivalent of these neuronexus arrays for putting into the non-human primate and ultimately to the human. This shows the complexity of doing these type of things on an animal skull. This shows the seizures that Anna L. showed you last week. And then ultimately this gets boiled into human electrodes that look something like this. This is our RC+S. So this is not that dissimilar from that responsive neurostimulator in that it records neural signal and stimulates, but is capable of higher degrees of analysis of doing spectral analysis and being able to look at biomarkers and so forth. Anyway, that's for the future. But another side of the lab, we've been asking the question now, can we have a more mechanism and neuron-specific approach? So electrical stimulation, we haven't talked about the mechanism at all. We don't really know how electrical stimulation really helps epilepsy or even Parkinson's disease. In fact, we as a field have been like a sine wave going back and forth between thinking this is inhibiting neurons and it's activating neurons. Maybe it's activating and inhibiting neurons. So we don't really know the mechanism of action of these things. So we're really interested in trying to figure out a more mechanistic approach. And instead of just sticking electrical wires into the brain and hoping that the right neural population has the right thing done to it, we are working towards more specificity. So we're using light for this, and I know that many of you are familiar with the approaches of Optogenetics, and Garrett Stanley is here, and he, of course, his lab, as you know, works very hard in this area. And so these optogenetic channels, just for those of you that don't know, they come in both activating and inhibitory forms where we can actually introduce sodium ions or chloride ions or protons and thereby specifically activate neurons using channel adoption or inhibit neurons using halo adoption or MAC. So there's a lot of different flavors of these things. I don't want to spend too much time going into the details. For translational approaches, as we are always focused on, we're not very interested in transgenic animals. You know, the likelihood of making a transgenic human is very low, especially since epilepsy usually presents later in life. The ship has already sailed on that. So we use viral vectors to introduce optogenetics into animals, and then you can put light guides in, light fibers, and achieve behavioral responses. So we started, actually Steve Potter and I started a number of years ago to bring this into the epilepsy model system. This shows you one example of one person who beat us to the punch by a long margin. Esther Cook-Magnuson showed a nice responsive system. I guess it's an on-demand optogenetic system. So just like that responsive neurostimulator, you can look at the EEG, as shown here, and decide when there's a seizure, and then turn on an optogenetic LED in animals that have been transfected. She used halorhodopsin to inhibit excitatory neurons and channelrhodopsin to drive inhibitory neurons. And you can see that when the light is on after a detection, the animal doesn't have a seizure. And when the light is not turned on, the animal has a seizure. Light on, no seizure. Light off, seizure. And this just shows amplification of this and a clinical response. So this actually does work. In fact, there have been a large number of papers now published on using light in optogenetic form in various instantiations for accomplishing this. In our lab, we're concerned with putting large amounts of light and light fibers into the hippocampus. So for patients whose hippocampus is doing something, putting in a single fiber is insufficient, and putting in multiple fibers is potentially damaging. So we've been looking at targets that are much smaller and that are easier to control. So the medial septal nucleus is one such area that projects its fibers to the hippocampus and is actually the pacemaker of the hippocampus. And it has various different populations of neurons, GABA, cholinergic, glutamatergic, and is the type of area that if you put an electrical microelectrode in or a macroelectrode in here, you have no control over what you're doing to this subpopulation of neurons. So it lends itself very nicely to being able to do cell type specific control of GABA, glutamatergic, and cholinergic neurons. So this shows a viral infection of the medial septum, and then nice expression along the septal hippocampal pathway all the way into the hippocampus. This just shows our approach to this of turning them on, turning them off, and this is work that's ongoing in the lab. Sang Park, who's a bioengineering student in the lab. So hopefully we'll have results to show you on that in the coming months and years. Now, the translational problem ultimately is a big one for me. So that optogenetics works in mice in particular, and even rats, is fine, but how do you get optogenetics into the human? Are we going to be in the near future putting light guides into patients? Now, I showed you putting a fiber optic into a patient's brain for destroying the hippocampus, okay? But are we going to be able to distribute enough light without causing damage into the hippocampus to be able to get optogenetic control? The problem is really depicted here by the fact that this is the amount of spread that you get in a rat brain, and the size difference is substantial between that and a non-human primate, not even to mention into a human. So this is the problem, the spread of light, and these are some of the solutions that are being promised and being developed, but we decided to take a different approach to get around the physical limitations of external light sources in the brain. So this is our answer to that problem. So light is produced by many other things aside from light-emitting diodes. For example, this sea of bioluminescent organisms provides copious amounts of light, and they provide it at various different wavelengths. So with Ken Berglund, who's an assistant professor in the lab, we've been developing bioluminescence coupled to optogenetic channels. So in the presence of substrate called coentyrazine, or basically luciferase, light is generated by an enzymatic reaction and then can be coupled ultimately to these to halorhodopsin, channelrhodopsin, what have you. So this is what it looks like in a depiction. This is channelrhodopsin, and this is its YFP, and this is Gaussioluciferase, which is introduced as a fusion protein to the channelrhodopsin channel, and then in the presence of coentyrazine makes blue light and can self-regulate its own channel. You can also switch the channelrhodopsin to just to point out that we can also, as a check, use standard optogenetic approaches, because this does not invalidate the approach of introducing external light, so we can compare the two. And then we can switch the channelrhodopsin to halorhodopsin, and make a different type of light. You can do this in the same cells if you want. So this is vanilla luciferase, which generates, with its unique substrate, yellow light to activate halorhodopsin. So what this looks like is this is a static fluorescence image. And then in cultures, let me see if I can get this mouse to come over here. You can see that in the presence of substrate, we get nice bioluminescence over that whole network. So again, these are the two different forms. We've called the inhibitory form of this inhibitory luminopsin and the excitatory luminopsin. This just shows the experimental implementation of this. And again, we can use these optrodes, microelectrodes coupled to optrodes as a means of using the conventional approach. We can put in multi-electrode arrays like we were doing before with the electrical approach. But here we simply use it for recording within the CA3 and the CA1 area. We've been able to show this works in patch clamp experiments. So you see the fluorescence and the luminescence, and you can see coenzyme. It generates a nice spike and a nice inward current. We ultimately are able to show that this can actually control rotational behavior. These videos are inactive, but you could see them rotating if you wanted to. We've quantified this, and this shows that with coenzyme, we can actually cause ipsilateral rotations when put into the basal ganglia, into the substantia nigra pars reticulata. And the nice thing about this is that you can actually see the luminescence in the animals themselves. So you can see the control signals being generated as we're doing the experiments. We're applying this in various different approaches. We've recently applied this in Parkinson's disease to show chronic activation of dopaminergic neurons by luminopsin. And we're actually looking at this in a neural restorative way, where by increasing the activity of axons, we can actually get BDNF, neurotrophic factor secretion, and have neuroprotective effects in these animals. I'll show that in a second. With Ling Wei and Xiaoping Yu, I'm sorry, and what's his husband's name? I forgot. Shanping. We're doing this in a stroke model, looking at transplantation of engineered stem cells combined with chronic excitation. And James Zhang is a graduate student in the lab that recently defended, showing this. And Miles McCrary is also doing that presently in Ling Wei's lab. And we're doing this in my lab with epilepsy. So this shows the rat experiments for Parkinson's disease protection, and we can see that with coenzyme, we're actually able to get a nice clinical response in these Parkinson model animals. This is the work of Jack Tung, who got his PhD in biomedical engineering here a couple of years ago and is at Stanford right now. And what he was able to show in cultures, these are these multi-electrode arrays that I showed you, that Steve Potter that really started the whole thing. These are the very same cultures on microelectrodes, and you can see in the ILMO, the inhibitory luminopsin expressing cultures, the firing rate is decreased as compared to control sister cultures, quantified over here. We then translated this into the awake behaving rat. And again, you can see the same thing, that in the presence of coenzyme, we get decreased firing in the hippocampus. This is a bicuculline model, not that dissimilar to the tetanus toxin model. And you can see that we're able to get decreased activity, decreased seizure activity. Again, what you see here is with a little bit of delay. This is in the vehicle, and this is in the coenzyme infused animals. You see a decrease in the number of discharges in the hippocampus. This shows then decreased in seizure activity. These are animals that are where the luminopsin, inhibitory luminopsin, is infused into the hippocampus or the dentate gyrus, and we can see decreases in seizure duration. What's interesting here is that the seizure duration decreased, but the actual intensity of the seizures didn't decrease when we just infused this into the dentate gyrus. The nice thing about the luminopsin approach is that we can infuse this in multiple brain regions. We're not limited by all the areas that we can put a light guide or fiber optic into. Any place that we can get gene expression by transduction, we can target for epilepsy. So we've been exploring now the idea of multi-site neural modulation. And so here he put this into the dentate gyrus, but also into the anterior nucleus of the thalamus, which is part of the circuit that involves the hippocampus in the so-called circuit of Pepez. And in that case, we're able to see decrease in duration as well as in, can't even see that on here, but well, in this particular, this is the anterior nucleus alone. I took out the slide. Sorry about that. This is the anterior nucleus alone. In the anterior nucleus alone, we got decrease in duration, and we didn't get a change in the Racine score or the intensity. But if I left the slide in, you could see when we coupled this together, we finally got decrease in seizure duration and in the Racine score as well. So these things seem to be additive. So luminopsins for epilepsy are nice because they allow more widespread neuromodulation than conventional optogenetics. There's a gain in the distribution, but one of the things we lose is temporal control. So instead of being able to flip the light on and off, like Cook-Magnuson was able to do, we have to rely on drug delivery. But what we're looking to develop now is there's been some really neat microfluidic devices for very tight drug delivery and controlled drug delivery. So we're thinking about beginning to try to apply this work where we can have great control of the drug delivery, and then we've solved both problems. We've got both temporal control as well as a better distribution. So we need to solve the substrate delivery hurdles. There are certainly pros and cons of the systems, and of course some of you may have heard of designer drugs or designer receptors exclusively activated by designer drugs. It's another chemogenetic approach like the luminopsin approach, which has certain advantages and disadvantages as compared to luminopsins. And we're working on autonomous responsive luminopsins, where we could provide the substrate continuously, but not have the luminopsin be activated, but building in a responsiveness into the bioluminescent part of the molecule. For example, putting calcium calmodulin and conferring response to calcium levels within the neurons to make an autonomous responsive luminopsin. And this hopefully will lead eventually to a completely non-invasive approach using perhaps vascular delivery of the viral vectors, vascular delivery, put in a drug pump. At least there's something for me to do as a neurosurgeon, and hopefully get more widespread adoption. So to summarize, I'm going to summarize this by saying what all of this is basically exemplifies is unmet clinical needs, which I and my physician colleagues can identify. Treating epilepsy is good, but not great. We can get to 65%. We don't often get to 100% seizure reduction. Patients want to be seizure-free off of taking drugs. So we identify unmet clinical needs such as these, and we marry this to clinicians, scientists, and engineers working together to make new therapeutic options. These are our targets. I've told you about some of the tools that we have, and all of this is basically what Entice's mission is on the Emory side. And then we marry together with the Neural Engineering Center, Chris and Garrett and Lena Ting and Rob Butera and others here on the Georgia Tech side. We really are one center. We formed as two separate centers because of the administrative aspects of working under two different universities. But we are one center really working together, and I think somewhat seamlessly, towards all of these advancements. And that really is the focus of the group together. So just to acknowledge the people that are involved, I have to acknowledge John Willey, who's a junior partner of mine who's been doing this with me, especially the laser stuff, in particular for the last I guess it's 7 years. Matt, who I already acknowledged, some of the other people working in our group, the Emory neurologists, and Babak and Anna L. Working on the monkey experiments, and some of the previous students I've already named. And of course, our funding sources have to be gratefully acknowledged. So with that, I will conclude, and if anybody has time to stick around, happy to take your questions. Questions? Yes, Annabelle.
So really interesting stuff, especially with the limb toxins. Do you have any data or thoughts about what could happen with prolonged use, like homeostatic responses to chronic activation or chronic?
Yeah, I mean, well, first of all, there's the substrate. Now, the substrate coentyrazine is a foreign substrate, so it actually, as opposed to DREADS, does not actually have any other targets. So DREGs are metabolized, or NPOs is metabolized, said that wrong, it's CNO. It's metabolized to clozapine. So coentyrazine might have some toxic effects. Actually, in Japan, they sell they sell bioluminescent lollipops. So we're not too worried about that. But yeah, the idea is not to chronically activate these systems. I mean, right now we're using the substrate, and it has to be substrate dependent. We don't know what the effects of long-term activation are. With electrical stimulation, though, there is some data on this, because while the NeuroPace approach uses responsive stimulation, there are other approaches using continuous stimulation of the hippocampus, and remarkably, it has pretty minimal long-term effects. We've been doing electrical stimulation for decades in similar patients. So I think there's the possibility of habituation. I founded a company called NeuroVista with some colleagues, and the idea was that we don't want to have these things be active long-term because the nervous system would habituate, and so we have to make these responsive. But we don't really have any evidence to actually support the adverse effect of long-term continuous stimulation in most settings. So we'll have to see.
Kind of along those same lines, I was thinking a lot about like neuroinflammatory responses
Right.
and like excitotoxicity for some of these treatments. I don't know like how that affects like when you're treating patients, because those are also sort of side effects of
Right.
epilepsy itself, but also if you're trying to avoid damage to some of the same regions with neurostimulation, how you sort of balance.
So yeah, I mean, this has been looked at, looking at various markers of microglial activation and so forth. And in fact, anytime we put something in the nervous system, there is an inflammatory reaction around it. And gliosis that occurs as well in a more long-term fashion. So these are important. As far as the inflammatory effects of neural modulation per se, we and others look at this, and we don't see very much with respect to that. In fact, there are certain scenarios where you can actually see an anti-inflammatory effect of neuromodulation. I think perhaps the reason for it is because we are working in the context of how the nervous system works ordinarily. We're talking its language. Certainly if you do things that are non-physiological, we certainly get into damage zones and inflammatory responses there. But as long as we stay within the physiological range and just feed those physiological signals in a non-physiological way, I think we're usually pretty safe with respect to inflammatory effects.
I know some people have to head off to class. Maybe we'll take any remaining questions offline. Thank you again, Bob.
My pleasure.